Go ahead and get started. We're going to begin. Welcome back, everyone. Today, we will introduce our DNA structure and function. We have talked about DNA already as one of our molecules of life, and so we'll expand on that. And for lab, we'll be talking about the cell's life cycle, mitosis and meiosis, so cell division. Okay. Before we get started, our chapter 8, will describe really protein synthesis. And so I want to make sure that we understand where we're going with this molecule of life. And we know that the Central Dogma of Biology, and if you guys recall, this is basically describing the way information flows in the cell, right? And now we know that it starts with the DNA, right? And it's transcribed to RNA. And again, we'll describe this in more detail in our next chapter. And translated to proteins. You guys remember proteins, polypeptides. And in terms of function, when we think about DNA and its connection to function, we know that the proteins are really, I like to call them DNA in action. So as we look around the room, I like to think of each and every one of us as like a sack of proteins in a sense. And so that will define you and I, what we look like, our behavior, and the particular functions, particular function of the cells that lie within us. And so I want you to keep that in mind as we're describing DNA and its function, as we introduce this chapter 7. Okay. So chapter 7 starts off with a sad story, and it's a story that we, well, most of us think, excuse me, I have to think about this. Yeah, 2001, maybe not. Some of us were probably still not born yet. Start thinking about this, but it seems like yesterday. And this is September 11th, 2001, when we were attacked, terrorist attack, and we lost a lot of people. And we were in search for people when the Twin Towers were hit. And James Smithington brought his police dog Tracker from Nova Scotia to a disaster site, and was able to find many survivors. Yet in doing so, suffered quite a bit. Is this police dog collapsed from chemical inhalation, burns, and exhaustion. The dog survived, but lost its limbs, and then 8 years later passed away. But was a very good tracker, and those characteristics, that information is embedded within the DNA, within its DNA, within the animals, the dog's DNA. And so the DNA was saved, was kept, and were used to make clones of this individual, of this dog. Soon after, Tracker was cloned. These canines for police and military forces in Korea began cloning. And in cloning, each cell's DNA, each cell's inherited DNA is from the parent individual, in this case, Tracker. And like its, its original, the original animal, it has that blueprint, it has that information that is able to be transcribed into a messenger RNA, and ultimately protein, which will define its abilities, its behaviors. And so DNA, we like to use, this as the master blueprint, as a definition for DNA. I like to also consider it as a recipe for all the proteins that are made within the cell. And those proteins within the cell will define the cell shape and overall its function.